bone biology research seminar series. So in the second round, the very first speaker is Dr. Rupesh Shrivastava. Dr. Rupesh Shrivastava is currently in All India Institute of Medical Sciences, New Delhi, in the Department of Biotechnology. Rupesh has done his PhD from National Center for Cell Sciences, Pune, with Dr. Mohan Wani and Dr. G.C. Misra. After finishing his PhD, Rupesh then went for postdoc at Columbia University Medical Center and Roswell Park Cancer Institute, New York. After completing his postdoc, Rupesh returned to India and joined he joined at Sagar University, Central University Sagar, as assistant professor of zoology. And then in 2017, he joined All India Institute of Medical Science, where he is now continuing his research. Rupesh has a very glorious CV. At such a young age, he has achieved a lot of laurels, including G.P. Talwar Young Scientist Award twice, which is quite commendable. And then he has a Science Communication Award in 2017 from Indian Science Congress. And I hope that given his productivity in the field, he will continue to receive many more prestigious awards. Rupesh's area of research is a very cutting edge, which is quite contemporary, and which is osteoimmunology. And to be more precise, his area is immunoporosis, quote unquote, which is the role of immunology, or to understand immunology of osteoporosis. And to just highlight one of his recent major contribution in this area is that he discovered and established the anti-osteoclastogenic role of B regulatory cells in osteoporosis. So with this background, we are eager to hear his research from his laboratory, and he will speak for 40 to 45 minutes, followed by some questioning, Q&A. And this time we are, with his permission, we have decided to record this presentation. So I now invite Rupesh to give his presentation.
Thank you, Dr. Chattopadhyay, for such a generous introduction of mine. I don't know. So basically, should I share my slide? Do I have the permission to do that?
Yeah, please.
Yeah, I'm just
you share it from your side.
Yeah. So, is it visible?
Yes, sir, it is visible.
It is visible. Yeah.
Fine. Okay. So first of all, I would thank again Dr. Nabedda for inviting me for this very prestigious initiative from his end, which is basically bringing up all the bone biologists of the country on a single platform. So my talk for today is basically something which I have been doing in the past, I think it's around 8 years being in India back from US. And the time since I came back in India, I started a very completely new field depending upon the situation and the facilities available to me at every step of my career. So this is just a summary of that, and this research is basically from the last, it will be covering almost around last to 5 to 6 years of my research work here at Sagar University and then coming to AIIMS, New Delhi. So the talk is something around the pivotal role of Breg, Treg, and Th17 cell axis in immunoporosis. So first of all, the most important thing is bone health. I know this is not a thing which should be shared here, but this I like this quote very much from the World Osteoporosis Day report. We have a single skeleton and which has been formed before we are born, and it supports us like anything throughout our lives, and it is going to remain with us long even if we die also. And regardless of the age, gender, race, nationality, or belief set, we all have only one. Yet this essential organ is so often taken for granted. So I hope this is the punchline, that this is a very essential organ, and then also we all are taking it for granted. We are not understanding how much important it is to take care of your bone health. So with this, so under bone health, there have been various diseases. So mine area of, or my niche is basically osteoporosis. So how I started osteoporosis basically, this is why I started osteoporosis. This is one of the scientific reasons. And one of the professional reasons is being that my training was done in such a way that I got to learn both immunology, hardcore cellular immunology, and hardcore bone biology at NCCS Pune under the mentorship of my guide, Dr. Mohan Wani, who is a superb bone biologist, along with Dr. GC Mishra, who is one of the best immunologists in the country. So I got this opportunity to be basically trained on both these independent field. And this is how I started working on osteoimmunology. So when I came back in India, I was searching how to start something which is quite unique and it's my own niche. So there I decided to start working on osteoporosis because it's a chronic bone disease and is characterized by very low bone mass and deterioration of bone tissues. And this is the deterioration leads to basically enhanced risk of fragility fractures in adults or in patients who are having osteoporosis. So just have an idea key, what is the global scenario? So we have around more than 200 people who are basically right now suffering from osteoporosis. So this is just an estimate. The current numbers will be quite high. So if you see that again, almost 9 million fractures, they occur annually and resulting in almost one fracture every 3 seconds. And both the men and the women are equally or almost equally been affected by this. The women's being slightly more, 1 in 3 women and one in 3 men are going to be affected by a fragility related fractures or osteoporosis in their lifetime. So this disease is going to hit each and every one of us. Even though we are not going to have a fragility fractures, we are at an enhanced or greater risk of developing osteoporosis, which can anytime lead to a fragility fracture. The only thing is that we have to just fall. So by 2050, the Asian scenario is not that quite good. The data suggests that almost more than 50% of the fractures will occur in Asia because most of the population is residing here in India and China. And whatever the Indian data is regarding osteoporosis, still it is not considered as a very a disease which should be given due credit, which it deserves. Still, we have to basically the funding agencies and the government have still to recognize this, that this is going to be quite high. So this is almost 25 to 60% of Indian women are basically at risk of developing osteoporosis in their lifetime. And the people who are at higher risk are basically the elder people and the women's, along with the men's also, although at a slightly lower risk. And if you see this data, the hospitalization data, so almost 43% of the hospitalization basically are occurring only due to osteoporotic related fractures. Only just 25% from heart and 26% stroke and only 6% from breast cancer. So if you see this data, that 43% of the hospitalization are occurring only due to osteoporotic fractures. And of these patients who are admitted, almost more than 25% are going to have a mortality rate. More than 25% of the people or the women or the men are going to die due to these fractures ultimately in a year. So the economic burden of this disease is somehow it's more than 130 billion USD. It's only the estimate of the Western countries, because in India we don't have that data. What is the economic burden of this osteoporosis, because people don't take it very seriously. And nobody actually goes to a health clinic due to osteoporosis. Nobody in basically, haven't seen anybody who's going in my own family also. We just have a screening for BMD and just to assess the risk of osteoporosis. We only go to the clinics when we basically have a fracture. So that's the time when we basically have already lost the chances of taking care of a bone. So this basically is why I chose this disease. And this is what we do at my lab, which is osteoimmunology lab. And these are the things which we do in our lab. So we have the following aims, which the first one being unraveling the nexus between bone and immune system, that is osteoimmunology, so that we can better understand bone with respect to various other organs also, and the role that is the host in host physiology in general of the bone, and how we can develop novel therapeutics and diagnostic strategies, keeping osteoimmune system at the core of this. We have various thrust areas. You can see here osteoimmunology, immunoporosis, osteomicrobiology, how microbiome and bone are affecting each other, nutrition, how diet and bone health are linked, how the environment and bone health is linked, osteotoxicology, osteofertility, how bone health and fertility are regulated, how we can develop various therapeutics. It could be probiotics, it could be phytotherapeutics, or it could be also alternative medicines such as homeopathy or Ayurveda also. How pathogens or the diseases basically are affecting bone health, and how various other inflammation diseases are also related to this. This is a general immunology which we are also interested in being at AIIMS. And also recently we also started this, what are the long-term effect of COVID-19 on bone health. So this is what we do here. So we are basically covering each and every facet of the link between your bone and the various physiological roles of bone in each and every organ, with each and every organ. So now coming again, I'm zooming in. So immunoporosis, which will be going to be my talk today, will be something related to immunoporosis. So what is immunoporosis basically? So I call this immunology of osteoporosis. And this is a term which we have coined and established in the field of 2018. And this was published that time with a lot of resistance from the scientific fraternity that time. They were very much resistant. All the reviewers were saying, why you want to start a new field when we already have osteoimmunology? The thing is again very clear that nobody understands osteoporosis. People think it's only an estrogen loss condition. It's not at all related with immune system. But this is not the case because some of the researchers have already shown various significant effect of immune system in osteoporosis, along with us, which we from the last 8 to 9 years having a number of times have proven this again and again that the role of T cells are very important in this balance of your pathological conditions observed in inflammatory bone loss in osteoporosis. So you can see here we have various adaptive immune cells and your innate immune cells, and all of them secrete some of the cytokines which are good or some of the cytokines which are bad. And ultimately, these all are basically there's a strong nexus between the bone cells and the immune cells, and ultimately the result is either a bone loss or a homeostatic bone balance. So this is the paper which received, I think, 96 citations till yesterday. I was just updating this, and more than 15,000 downloads, 3,000 downloads and 15,000 views. And now this field is quite established. I can say this because if you search on PubMed, more than 6 to 7 papers are there, apart from my own lab, which are taking immunoporosis as a established subject nowadays. So apart from this, we also have one bone reports paper, which was highly cited, which is among the most cited papers of bone reports till now. How we have shown how the lactobacillus acidophilus is basically regulating your bone health via this Treg and Th17 cell axis, that is again immunoporosis. So this is all we did, and we were basically recognized by Immunity, and we were on the cover page of 25th anniversary issue of Immunity Journal in April 2019, and my photograph was also there. It's very small, we cannot zoom in right now. So this is how they recognized our efforts of contributing toward this important field and establishing this field. So now, again, so this is a very important axis, which is an immunoporotic axis. And if you believe me, and all of you must be aware of, that this is the axis which is being exploited by all the available drugs. FDA approved. Only 3 drugs are FDA approved for this as a therapy for osteoporosis. On all these 3, basically the bisphosphonates or the teriparatide and the denosumab are somehow directly or indirectly targeting this immunoporotic axis. Either they are targeting something which is secreted from the immune cells, this is rankle or your inflammatory cytokines, or inhibiting osteoclastogenesis, which is already been regulated by the strong immunoporotic axis. But the problem is all these three drugs, which are the only available therapies for osteoporosis, they all have serious side effects. The most latest one of them, which has been reported, is depression, which is just 2 months back only. They have reported that long-term use of these drugs lead to depression in women's. Apart from that, you have serious heart attack risk also, along with developing breast cancers and osteonecrosis of jaw and ulcerations also. So that's why, this is why, even though we have these drugs, we still can't sit and relax. We need to find certain drugs which are more effective. Apart from that, being more safe also. So this is how we started our research, the quest for searching a novel therapeutic strategy, which is quite effective also. And apart from that, it is also simultaneously very safe also. And this is how, after coming back to India, my PhD training was in rheumatoid arthritis. So there I worked on Treg and Th17 cell axis, how that was being regulated and how IL-3 was basically tweaking that axis and leading to your amelioration of the disease. So I thought, and when I searched the literature regarding osteoporosis, what is the condition with osteoporosis, I didn't find each and not a single paper which has been reported how this Treg and Th17 cells, what is the state of these cells. Then I decided, okay, this is a very strong axis and we should look into this. And this is how it started working in this important field. And we all know that under any inflammation, inflammatory bone condition, this balance is tweaked. Example in RA also. And this is how we basically took probiotics, and we have shown this by various papers in the last 3 to 4 years that Lactobacillus rhamnosus, Lactobacillus aerocervilis, they all are modulating the Treg Th17 cell balance, and they are maintaining your bone health, and they're also increasing the bone heterogeneity also, again an important factor in your clinics. And this again, Bacillus clausi also, we have shown that. And apart from not only taking probiotics, also we have shown how high dietary salt intake is basically via this axis can impair your bone health in case of your osteoporosis, and also how environmental pollutants such as MCLR, which is cyanotoxin, is also basically detrimental for bone health in case of your osteoporosis and inflammatory bone loss. So this axis is now quite well established. So now these are just a peanuts if you see the immunological arsenal of the cells which you are having in the body. There are very small number of T cells and are there. Apart from this, we are having various n number of other cells which we don't know currently and which we still don't know. For example, ILCs, which have been discovered in the last 5 years only. And before we have known about them, but now since we know about them, we are thinking that each and everything can be also governed by ILCs also. So we were looking into these can't be the only cells in the immune system which are regulating bone health. So then I started searching for there should be some other cells also, or maybe most of the cells will be affecting this axis somehow. Then I started, what other than T cells? So the first thing which came in my mind was B cells, because T cells, B cells, we normally say this like this also. B cells again, because I was working on B cells also for some months in my PhD work, and that why B cells were also very close to me. Then I searched for B cells, and we also know from the literature that B cells are secret rankle, and they have been involved with both positive and negative effects in case of various bone pathologies. So this is how I thought of doing something on these B cells also. So, but there are n number of B cells, if you see that. And recently, if you search the literature, you will see that there has been a novel subset of B-regs, regulatory B cells, which you call. They have also been discovered. So these B cells, if you're not talking about the regulatory B cells directly, if these B cells, if you take these B cells alone, they have a lot of non-humoral functions. Non-humoral is a key point here. Why I'm emphasizing non-humoral is that we all know about the humoral functions of B cells. That is antibody dependent, because we only learn about the humoral functions in the immunology textbooks and the classes. What we miss is that the B cells also have a very important number of non-humoral functions. And I was searching in literature, I was just amazed how these B cells can also basically regulate your tissue development via your lymphotoxin alpha-1 beta-2, OPG, rankle, and how the galt and the Pierce patches, the the tertiary lymphoid organ, they all can be basically the tissue development is being regulated by B cells. And this is purely not dependent on the immunoglobulins, that is the antibodies. So this comes under non-humoral function. This is why very important functions of B cells, which is now taking pace with the research which is being done around the globe. How these B cells are affecting, are having a very important non-humoral functions. Apart from that, they also promote Th1 differentiation. They support Th1 differentiation polarization of macrophages also. They increase IL-12 production by DCs, and they also support T follicular helper cell development, Th1, Th2, and Th17 cell responses. So these are B cell non-humoral functions of B cells. But if we talk about regulatory B cells, which are named as regulatory B cells in 2003, I think, 2000, I think, by Mizoguchi groups. And these in the last, if you see 20 years are now, after the discovery of Bregs, if you see, they were initially called as suppressor Bregs. The concept of suppressor B cells were there, then later on it was converted into regulatory B cells. And from that time till 20 years till down the line, we still don't know much about these regulatory B cells with respect to various diseases. The most intriguing point was that, the striking point was that when I joined NCCs, there was a paper from Dr. Mishra's group in 2003 that if B cells are activated by LPS, by the TLR4 ligand, these B cells, if you activate these B cells with the help of LPS, then they basically express TGF beta on the membrane. And these Bregs, these B cells, not the Bregs, these B cells were basically able to induce NRG in CD8 positive T cells. So NRG is a state in which there is unresponsiveness, a state of unresponsiveness where the T cells, the CD8 T cells basically cannot or will not be able to respond to any of the antigens thereafter, after they have been co-cultured with these B cells. So this was very important because this was a function. When I joined the lab in 2006, I discussed with sir that these are regulatory B cells. In fact, these are not B cells. These are regulatory B cells because they are regulating the functions of CD8 T cells. Then he agreed with me, but they haven't named it as regulatory B cells. So 2 years back, we wrote a review article and we have named that regulatory B cells now. And that is a novel subset of regulatory B cells, which we call as BR2 Bregs. So if you now see, the Bregs have various role in immunity in general. They have inhibitory effects on various immune cells, and they also have promoting effects of these Bregs on Tregs, on regulatory TR1 cells, DCs, and induced natural killer T cells also. These are just some of the functions. Important functions have been only shown here. So they have a role in various inflammation and diseases, starting from COVID-19 to the bone, which we discovered recently, and this is what I will be discussing here today. So they have functions in allergy, asthma, graft rejections, autoimmune diseases, and inflammation associated with disease parasites, along with inducing tumorigenesis also. This is one of the functions which is basically not good function of regulatory B cells, just like the Tregs. They promote the growth of cancer and tumors. So since we are now going to talk about Bregs, I will give you a brief idea about what are the various subsets of these Bregs are there in case of both mice and humans. So this is the BR2 Bregs, which I was talking about, which we have 2 years back only we have reclaimed and named it again as BR2 Bregs in a review article. And today I will be focusing on these B10 Bregs, which are basically IL-10 secreting. They all basically, these B cells, Bregs are basically IL-10 secreting, the major signature cytokine of any regulatory B cells, either in case of mice or humans. So I will be talking about this particular subset of Bregs here today. We are also working on this, but the data of this are slowly developing. So we will leave this for now and we will be focusing more on this B10 Bregs. So the first question, which is how we generated Bregs. So the population of Bregs is very, very minuscule in the body. It's less than 1%, I think. So in spleen or maybe in the periphery, it's very hard to find. So if we are going to do something in vitro, if you're going to some research, you all know that we should have that particular cells or the molecules in hand in a sufficient quantity. So we have to generate Bregs. So how to generate Bregs? That was again a very tricky question because all across the literature, there have been various protocols for generation of regulatory B cells. So what we planned was that we isolated the B cells from the mice with the help of negative selection or flow sorting of these. And if you see, we enriched these B cells, more than 95% purity was obtained. And then we stimulated these B cells with LPS at 10 microgram per ml. We took LPS because LPS is a very well established method in all the literatures available when we searched. These were the one which was quite robustly inducing the induction of regulatory B cells. And you can see here yourself also that your induction with LPS is 53 times inducing the generation of IL-10 secreting CD19 positive B cells. So these are basically your CD19 positive, IL-10 positive regulatory B cells. And apart from that, we also have one more phenotype, which we call as beaten Bregs, which is CD19 positive, CD1D high, CD5 positive, and IL-10 positive. You see that CD1D high and CD5 positive, you see from 5.17 in the control, it's going up to 15 to 3 times induction of these population is also observed in LPS, apart from you having 25 times the induction of IL-10. So then after establishing this, standardizing this, the generation of Bregs in vitro, then we ask the question, do Bregs really have any anti-osteoclastogenic potential? So for this, to prove this, we put up the co-culture assays with the monocytes with the osteoclast precursors. We all know how we do it. We took the spleen, we minced it, we negatively selected the B cells, we activated them with LPS for 24 hours, and simultaneously, in parallel, we also harvested the bone marrow cells, cultured with MCSF overnight, and after 24 hours, took the non-adherent cells, counted them, and we co-cultured these Bregs, LPS-induced Bregs, along with these your precursors of the osteoclast in the presence of MCSF and rankle for 4 days, and then we performed the trap staining to assess what is happening. So we were very much excited after the results came in. These are the B cells. These bone marrow cells were cultured only with B cells, not the Bregs. These are the control ones, and this is the M plus R, how the osteoclasts are being generated without B cells, with B cells. And these are the regulatory B cells which have been cultured in the ratio of 10:1, 5, and then 1:1. So you can see here from as we increasing the number of Bregs, there is significant inhibition in the osteoclastogenesis as observed with the help of this trap positive multinucleated osteoclast. If you do some quantitative parameters also, you can see the number of trap positive cells are significantly reduced along with the number of cells with more than 3 nuclei, and the area of osteoclast is also going down significantly. So this particular experiment, the first experiment, was very interesting and very morale boosting for us. Okay, that's really great that Bregs have anti-osteoclastogenic potential, because this was not shown anywhere in the world just like this. So we then assessed are they functional or not. So then we did the F-acting, the same methodology we adopted. Just the only thing was that we stained them with phallodoin, and we found that these beautiful F-acting rings were observed in your cells of the osteoclastogenesis, and you can see how this is inhibited the formation of these osteoclasts, and the F-acting ring has also been distorted and diminished significantly in case of your cultures with BL these Bregs in a dose-dependent manner. So now coming, the next question was how. So we proved this that the regulatory B cells were actually inhibiting osteoclastogenesis. So next the question is, how are they basically inhibiting osteoclastogenesis? There need to be some molecule for that. And we all know that there are a number of molecules through which Bregs can work, IL-10, 35, TGF beta, or maybe something else also. So what we did, the first experiment, what we were interested in is that, because whether it is a contact dependent or a contact independent manner. So discern this, what we did, we again co-cultured Bregs with bone marrow precursors, but this time in a transwell setup in which the transwell was having the LPS activated B cells, that is the Bregs, and the lower chambers were having your precursors for the osteoclast. And we did found that after keeping these both cells apart with a barrier in between, the results were the same, that the Bregs were inhibiting completely. You're in a ratio dependent manner, it was significantly inhibiting your generation of osteoclasts. So this again proved that, okay, there is something which is contact independent, and we should look for that only, not the contact dependent thing. That is TGF beta. The role of TGF beta here was gone, because when you activate the CD8 B cells with LPS, they do have a membrane TGF beta expression, and that particular thing was basically we crossed that mechanism of action. And when we moved, then we targeted IL-10, because IL-10 is the major cytokine which is being secreted by all the regulatory B cells, irrespective of the phenotype in mice or humans. What we did to prove this, we did anti-IL-10 neutralization assay, and we did found the moment you add, so this is a M plus R positive control, and we put anti-IL-10 10 microgram per ml for catching up all the IL-10 in the media secreted by the regulatory B cells, and here we put the isotype for IL-10, anti-IL-10. The moment we added anti-IL-10, all the abrogative or the inhibitory effect of Bregs were gone completely, and it was coming back to just like the normal cells, M plus R control cells. So this particular experiment very beautifully explained and proved again that it is IL-10. So Bregs inhibit osteoclastogenesis in an IL-10 dependent manner. And we also did the functional assay also affecting, and the results were the same. Now, so this all was done in vitro. So now we were thinking, what is happening in vivo? So what we did, we developed the ovectomy model, we developed bilateral ovectomy model, and we did various parameters analysis, bones, serum, bone marrow, and spleen. We took spleen and bone marrow because bone marrow is the site where osteoclastogenesis basically occurs. So we have to look at bone marrow, and apart from that, spleen also, because spleen is the site where Bregs or the B cells are basically being generated. So if you see here in the control, the sham, it is 4.58% of the CD19 IL-10 positive cells are there, which is highly significantly reduced in ovectomy mice at both the bone marrow and the spleen. And the other population, that is the B10 Bregs, the CD5 and 1D high population, is also basically significantly reduced. So this particular experiment clearly proves that regulatory B cells is very important in case of ovectomy induced osteoporotic preclinical model of osteoporosis. So this we published 6 months back in Frontiers in Immunology, and this was the first report. Bregs actually inhibit osteoclastogenesis, and they have a very potential role in your ovectomy-induced bone loss. And with this, we also calculated that almost 1 Breg is equal to inhibit almost 30 osteoclast precursors. This we calculated at 1:5 ratio, but 1:1 it will be almost 5 times. So it will be 150 osteoclasts. 1 Breg will be inhibiting 150 osteoclast precursors. So that's a huge potential. If you're just having 5 Bregs, you can inhibit. If you're taking 150 osteoclasts, then 5 Bregs will be sufficient to hundreds of osteoclasts will be inhibited. That's really a very high potential of the anti-osteoclastogenic potential of these regulatory B cells. Apart from these, the subsets which we describe in vivo, we also discovered these two other regulatory B cells, or the 19 positive CD11B positive regulatory B cells in bone marrow and a spleen. They also were highly significantly reduced in case of ovectomy mice, and this CD19 positive FOXP3 positive Bregs. So we are still working on how these Bregs are also important and how they can be also contributing towards how the collectively all the Breg subsets, what are the Breg subsets, all the complete profiling, we are now doing that. And we will be coming up with some interesting data in due course of time. So now, after proving that we have this Treg and Th17 cell axis is already there, already proven by our lab, and after discovering that there is a role of Bregs in basically having an anti-osteoclastogenic potential and having a role in ovectomy-induced mice, we thought about there should be something called as Breg Treg Th17 cell axis. Why Breg I have kept before Treg is that Breg have already been known to regulate enhanced induction of Treg cells and inhibit induction of Th17 cells. So they are higher. If you see the hierarchy, they are at a higher position. They can regulate both Treg and Th17. That's why I call this Breg Treg and Th17 cell axis. So how? Or can? So under inflammatory conditions, bone loss, this axis is tweaked more towards the inflammatory type of Th17 cells. But if you give certain therapeutics, for example, probiotics, which we have been doing in the lab, they used to bring back this axis towards homeostatic levels, more of a Treg cells and less of Th17 cells. Can Breg also support this regulatory balance by further robustly supporting this anti-inflammatory environment and inhibiting the development of Th17 cells? This was the questions which we started looking after. So this was the question. So can Breg Treg Th17 cell axis be exploited as a therapeutics in osteoporosis? So this is what we then further looked into. So for that, we basically took a new probiotic, that is a Bifidobacterium longum. Why I took probiotics? Why as a therapy? Basically, I'm going to show you how by exploiting this axis with the help of any modulators of you as a therapeutic molecule. For example, here we are taking probiotics. We can basically enhance bone health. So the first question which came, why I have taken BL? BL because Bifidobacterium longum is one of the first bacteria which inhabit the infant gut. And it is also very much, the population of these BLs are very high in case of your human gut also. Along with this, the BL species is one of the species which have been the most extensively researched in the research community till now. And plus, they have a very strong immunomodulatory potential. So since I was going to do something on immunoporosis immunology, I was searching for certain probiotics with a strong immunomodulatory potential. So we chose this. What we did, the first question was that can it Does it have any anti-osteoclastogenic or some immunomodulatory potential or not? So what we did, we cultured this BL, and in anaerobic conditions, we took the BL conditioned media, that is a cell-free conditioned media. The pH was neutralized and it was filtered so that we can use those culture supernatant, the conditioned media, on these osteoclastogenic assays along with your Tregs and Th17 and Bregs assay, which will be coming up down the line. The first thing was that the first results are very interesting, that yes, BL was having that anti-osteoclastogenic potential. In a dose-dependent manner, it was able to inhibit the development of osteoclast, and the functional assay was also giving the same story, that it was not only inhibiting osteoclastogenesis, it was also functionally also it is making them the osteoclast incapable. Along with, so these, and the conditioned media was not at all toxic because we did the MTT assays along with your magnesium PI. Also, we didn't find any major differences between your control and these conditioned media supplemented cells. So like next comes, do these BL is also having effect on Breg, Treg, Th17 cell axis, because we will be connecting these cells later on. So yes, they were having these effects. So again, we took the conditioned media and we put those in your induction media for Th17 and Treg cell differentiation along with the Bregs also. These all three were done separately, not in a single well here as represented. These are just diagrammatic representation. So we did three assays, one for Breg induction, one for Treg induction, and third for Th17 induction. Under polarizing condition, that is, for Tregs, we added TJ beta and IL-2. For Th17, we added TJ beta, IL-6, IL-23. And for Bregs, we took the same LPS-induced Bregs. And on all these three conditions, we added your BL conditioned media. So the results were quite, again, very interesting. If you see here, we followed this gating in strategy, and we looked into what is happening to the CD4 positive and IL-10 positive cells, which we call as regulated type 1 cells, which are just CD4 and IL-10. You can see the induction, 8.68 to 44%. Such a significantly enhanced induction of the capacity of this TR1s are there under polarizing conditions. And if you see the FOXP3 positive Tregs are also from 8.9 to 18%. So that is again is quite high. And the IL-10 production is also quite high, so 4 times from 10.33 to 40.2% the induction. So such is the strong, significant induction of these TR1, Tregs, and IL-10 positive Tregs via the BL. So this is basically, it's very important. Then again, we looked what is happening to the Th17 cells. So again, if a good molecule should actually enhance Tregs and suppress Th17 cells so that the balance, the axis is basically taken care of. So again, the CD4 or gamma T, if you see from 15 to is coming to 4% with the BL conditioned media, if you're adding this, and also the IL-17 positive, the induction of these IL-17 secreting cells is also greatly, significantly reduced from 28 to 14, almost half here. And again, all these are just the quantitative data of these dot plots only. And third, what is happening to Bregs? Because these are the master regulators. Because Treg Th17 we already know having a role, but what about Bregs? Can they also do it? Can the BL also induce Bregs? So we took again these Bregs with 24 hours of LPS treatment and added BL also in them. And this is the data which would be very much interested in that. If you see here CD5 positive, CD1D high, the cells are almost quite enhanced with LPS 14% and LPS of BS 17.7%. This is quite significant because if you say 10 from 10 to 13, it is there. Although it appears very slow here, but this is quite significant. If you see the MFI of the IL-10 being secreted, that how the single cell is secreting how much amount of the IL-10, this is quite high. So this is a 30% increase in your induction of your regulatory B cells with your BL condition media with respect to your LPS control. So LPS is also giving us quite high induction of Bregs, but if you add BL, that is basically again almost 30% it was enhancing the induction of regulatory B cells. So the next question was that can these BL induced Bregs modulate the ostomy system? Because now we have, till now I have shown you the how BL is directly regulating your Tregs, Th17 and Bregs independent, completely independent setup in your in vitro. But now we were interested, can this BL induce after inducing the Breg generation with BL? Because if you see from this data, that BL induced Bregs are also quite significantly high being induced of IL-10 production with respect to just only LPS induced Bregs. So this data basically very much pushed me. We should look into how BL induced Bregs should be behaving in the osteoimmune system. So then what we did very smartly was that we cultured these Bregs with BL induced Bregs, which we will call with BL induced Bregs, along with the control Bregs just being generated in the presence of LPS. And we co-cultured these two Bregs, one only Bregs cultured with LPS and the other one LPS Bregs plus along with the BL with the bone marrow cells, precursors of the osteoclast in presence of NCS of rankle. And what we did find was that either traps, both the trap staining along with the Treg and T17 assays were done. And we did find that if you see here, if this data, we just focus on here on the Tregs. So if you see CD4 FOXP3, from 1% it is going to 4.2%. So this is very interesting. So one thing which I did was that these BL induced Bregs were co-cultured with naive T cells, both under polarizing and non-polarizing conditions. So under polarizing means we added TJ beta and ILT and all those things. Under non-polarizing, you just took these Bregs and cultured with naive T cells without adding any of the TJ beta, IL2, 6, 23. So that we should look at the power of Bregs in inducing either Tregs or inhibiting TS17 without any factors. Naturally, what is happening within the your in vivo, just replicating those processes here itself. So if you see the data of the polarizing conditions, I'm not showing you here because they are similar. The data of non-polarizing conditions, just culturing 90 cells without any added factors for Treg and TS17, what we found was that these Bregs, the BL-induced Bregs, were 4 times having the capacity of potential of inducing Tregs. 4 times. If you see for just for the CD4 and FOXP3, and the IL-10 was quite high, against from 7 to 11% was going. So this was again the case for T17 also. So we were having from 0.45, it was going down to 0.095% for the CD4 or gamma T, and also the 17 also, if you see 0.58 to 0.17. So for the potential, and also if you see the Bregs which have been co-cultured with the osteoclast, only the Bregs LPS activated and LPS plus BL. If you see, these are quite significant inhibitors. So the BL induced Bregs are having a very high potential of regulating the osteoimmune system. So almost 3 times they were inducing the development of Tregs. All 6 times they were inhibiting the development of TS17 cells, and 3 times they were inhibiting the development of osteoclastogenesis with respect to Bregs without any your BL stimulated. So BL stimulated Bregs were quite high, were having a very high potential in modulating the osteoimmune system. So once we establish this, so we also interested in what is happening. So this is some of the calculation which I made just to show you how what is the potential of these BL induced Bregs. So normally what we reported is that one Breg is sufficient to inhibit 30 osteoclast precursors. Literature suggests in 2007 from Zeiss et al. Group that one Treg is having potential to inhibit 100 osteoclasts. Okay. And we also shown here that Bregs were able to induce Tregs almost 3 times, 3-fold induction of Tregs. Okay, so ultimately you can say one Breg would be able to inhibit 330 osteoclasts, 300 from here and 30 from here. So more than 330 osteoclasts can be inhibited by just one Bregs. But when you are using BL induced Bregs, this is just for Bregs. The above was just for Bregs alone without any BL addition of any BL condition media, the probiotics. If you add the probiotics here, you can see that capacity is increased, almost doubled from 30 to 60. And this your one Treg is almost 100, that is already known. And the the Bregs are 3-fold induction, BL induced have 10-fold induction. So again, so this is almost coming to more than 1000 osteoclasts. So one BL induced Bregs is having the potential to inhibit almost more than 1000 osteoclasts. So this is a very huge, basically upscaling from Bregs alone to BL induced Bregs. You're almost from 300, you are going to more than 1000. And this calculation has been done taking the data of the cultures with 1:5, one Breg with 5 osteoclasts. If you take 1:1 ratio, this will be again 5 times, more than 5000. So this is the potential, the strong anti-osteoclastoid potential of these BL-induced Bregs. This is basically, again, all this data have been done in vitro. Then we were interested in what is happening in vivo. What we did was that when we again did ovrectomy experiment, we developed the ovrectomy-induced osteoporotic mice model. And the third group, we added BL. We give them BL daily for 45 days and sacked them and did all the osteoimmune parameters. The first one was the SEM analysis. You can see here sham, the cortical bones. If you see the surface of those are very not very bumpy, but here you can in the ovrectomy you can see a lot of pits are there, your resorption pits are there, which are basically correlating with the enhanced osteoclastogenesis under these osteoporotic conditions. But the moment you add, you feed them BL, this is again going back to the near normal levels. And this is the MATLAB analysis of these things, showing the correlation between this bone mass and your bone loss. And the red one represents the higher correlation and a better condition of these BL-induced ovariectomy, BL supplemented mice. These are the microcity analysis we did for the trabecular LV5, which is the gold standard for your assessing your BMD parameters in osteoporosis, along with the femur. Also, you can see the how BL was basically enhancing bone health by inhibiting bone loss. So apart from this, the BMD data was quite interesting. They were able to enhance the BMD in all the five trabecular, LV5, femur, tibia, femur cortical, and the tibia cortical. The BMD was significantly enhanced. And the bone strength parameters were also very, data was very encouraging, that the power was increased, the energy was increased, and even the stiffness was quite better in the BL supplemented mice. Now coming back to what is happening to this Breg-Treg-Th17 cell axis in these in vivo mice, because all data we have established till now was in vitro. Treg-Th17 we already know that it should be is bound to increase, and this is how this is happening. So this is how this is happening. If you see here, you sham, OVX, and ovectomy plus BL. You can see here in the bone marrow, spleen, MLN, and PS patches, CD4 positive and FOXP3 positive. This is how it is happening. In the ovectomy, it is 10%, and ovectomy plus BL, it is again coming back to near normal levels of the sham in almost all of these lymphoid organs. Bone marrow is the primary one because here only osteoclastogenesis is occurring. The Th17 cells are also inhibited. Here significantly is going down. In case of rectomy, it is up, but with BS supplementation, it is going significantly down. So the Treg-Th17 axis is being taken care of. But what is happening with the Brex? So this was important. And the Brex also, if you see, we have already this data from the sham and OVX. We have already shown that in frontiers immunology paper that in the ovariectomy, it is going down, and in the sham, it is high. But if you see with the help of BL supplementation, this is again coming back to near normal levels. So this is again basically you're increasing, enhancing your regulatory B cells. The BL supplementation is basically doing this. So this is how these data basically strongly supported our hypothesis that there exists a Breg-Treg-Th17 cell axis, and if you can tweak that, we will be able to take care of these bone loss conditions under osteoporotic conditions. So now coming back to here, what is happening, the serum cytokine data also, if you see the IL-17 and the IL-10, these are basically the signature cytokines, for IL-10 being for both the Tregs and Treg and Bregs, and IL-17 for your Th17 cells. So this is how your Th17 is going down and how the Bregs and the Tregs both are coming up. And looking into the in vitro data, how Bregs actually stimulate Tregs and inhibit IL-17, this data is quite very interesting that yes, there exists a Breg-Treg-Th17 cell axis in case of your osteoporosis. So this is again, I'm just summarizing this. Under osteoporotic conditions, estrogen deficiency, these Bregs are not at all induced or very not that much induced, I would say. I would reframe my word. And these Bregs are already known to induce Tregs and inhibit Th17 normally also. But what happens in under estrogen deficient conditions? The stimulus is gone and there is completely 360-degree overturn is there. But what 180-degree overturn is there? Sorry for that. So what happens here is that with the treatment of Bifidobacterium longus supplementation, this balance is again coming back directly also via the BL supplementation, directly on the Bregs, Tregs, and Th17 inhibition is there. Apart from that, there is also an indirect regulation via the Bregs. So this is how your Breg, Treg, and Th17 cell axis do exist in your osteoporosis, having a very strong role in regulating your osteoporosis and inflammatory bone loss. So these are the clinical applications basically which we can target and which we are doing actually in our lab here, that we are taking up the osteoporotic patients and doing these all BL condition media experiments on them. This is just one of the control experiments I'm showing you because the other data still we haven't communicated. So and still we are developing that. So that could be part of some other talk, somewhere else. And the point is, so if we somehow increase a 10% of BMD also, if you can increase the BMD by just 10%, we can delay the development of osteoporosis by almost 13 years. So this is how the aim should be there. So even a small increase of 10% would lead to delay in 13 years, delay of 13 years for development of osteoporosis. And probiotics have that potential to do this, which can be just taken as a nutritional supplements daily, and it would be basically a game changer in treating and managing osteoporosis. So the key is basically osteomicrobiology. How this therapeutic key can be used for exploiting this Breg, Treg, and Th17 cell axis. If you see here, what happens under normal conditions, that the basically under normal conditions, everything is normal. The normal number of Treg, Th17, and Bregs are there, and Th17s are being taken care of. The regulatory reasons and Bregs. Under inflammatory bone loss condition, osteoporosis, RA, there is a leaky gut. So there are a lot of inflammation is there, leading to your enhanced Th17, less number of Tregs, less number of Bregs. The balance is shifting towards more of osteoclastogenesis and less of osteoblastogenesis. But if we modulate this gut microbiota with the help of either probiotics, prebiotics, symbiotics, or fecal microbiota transplant also, or better using some SCFAs or gut-associated metabolites, basically we can tweak this balance and bring it back to near normal levels. So this is now coming back to my last slide almost. So these are the contributions of my lab. So we have established the gut immune bone axis in osteoporosis. We have used probiotics and harnessed the potential of probiotics for treating osteoporosis. We have established the Treg Th17 cell axis immunoporosis. We have discovered the immunoporotic role of Bregs. And lastly, we have now established in the phase of establishing the role of Breg Treg Th17 cell axis in immunoporosis. So these are my selected publications from the last 3 to 4 years. And thank you, all of you. So most of the work has been done by my PhD scholars here, basically Lina here. And these are the funding agencies. These are funding agency, Department of Science and Technology funded project. This is basically the Department of Science and Technology funded project, this particular, and these are the collaborators of mine. And a special thanks to CDRI and Dr. Nabedya for providing me the opportunity of doing the microcity analysis and the bone strength analysis in his lab. And last but not the least, to the mice which have sacrificed their life for such a wonderful study and for the mankind. And lastly, we should love our bones because we just have one and protect our future. And this ends my talk here. And I would be happy to take any questions now.
Thank you very much, Rupesh, for this wonderful talk, and congratulations on the body of work and the contribution to this field of immunoporosis. You have given so much significant contribution in such a short time. I hope that you continue to remain so productive and give more and more insights to immunoporosis. There are many people. Those who want to ask question could raise their hands. As you prepare for asking question to the speaker, let me clarify a couple of things, Rupesh, from you. Number one is that you have your data is quite unequivocal in demonstrating that IL-10 from these B cells or the Breg cells are crucial in inhibiting osteoclastogenesis. Now, there is also, or there are reports that in the bone marrow, it is the B cells which are the major sources of osteoprotegerin. So, is there any possibility that a certain subtype of B cells from the bone marrow, through elevation of osteoprotegerin, would result in suppression of osteoclastogenesis?
So there is a strong possibility of this, and we haven't done this OPG-Rankel ratios in the serum cytokine analysis, but we will be doing that. We currently are doing that, and this is a very, very important question because I already know that. Plus, one more thing is there. To clearly prove the role of Bregs in osteoporosis, we need certain knockout mice, and that are missing from AIIMS. We don't have such kind of facility here, and that was the only reason we were not able to basically communicate this to a much higher journal. and we have to compromise on this because we didn't have that facility, and we cannot wait for that also because this is such a competitive field. That's just we want to communicate and take this notion to the world that Bregs have anti-osteoclastogenic potential. And yes, Bregs, the B cells are already known to secrete OPG also and rankle also. And the ratio of these depends upon the condition of the inflammatory condition or the disease state of the mice also and the human beings also. And this is a very nice question, and I'm also planning to do this. How basically if Bregs are also secreting OPG in a higher amount, that would be very wonderful to do that. So I will be taking note of this, and maybe I will be looking into this also in my future experiments. So how if Bregs can also basically are having a higher amount of secretion of OPG, so that will again give a very strong impetus to this Breg field of in osteoclastogenesis.
Another thing which is actually after seeing your very provocative data on the ability of individual B cells or the Breg cells or the BL stimulated B cells, that almost 1,000 osteoclasts can be inhibited. Now, if you are able to do an experiment such as this, I'm just curious to know, suppose you want to do, if you just simply do overectomized mice, how many, if you count, will 10 BL activated B cells be sufficient to completely prevent its bone loss?
So this is this is I was expecting this question from someone. So basically we are counting that, and we are now planning to transfer the Bregs, okay, in this ovariectomy mice. So we are planning this, and maybe in the another 5 to 6 months we will be able to do it also. The only problem is that getting sufficient number of mice to induce these Bregs and then transferring them to these ovariectomy mice, again keeping all those your power calculations. So we are not getting that much number of mice here in our facility. So we have that kind of thoda sa we are having that crunch in number of animals. So that's why we are not able to plan it. Once we thought of planning, once we also did it, but the thing was that we didn't get the number of mice required for taking out these Bregs and the naive B cells and then inducing into Bregs and then transferring into those. But we will be doing this. We have now again looked into how to increase the number of these animals in the colony, and then we are planning to do this. And after that only I would be able to give you certain clear-cut idea ki how much number of Bregs. So this is what I'm pitching for. Although it is going to be very costly and it's not going to be very economically feasible for the patients, we have a Breg therapy for osteoporosis. But yes, that will prove a concept that these Bregs are having such a strong potential. The probiotics can modulate them 5 times with respect to the Bregs. And these Bregs, basically, I'm also having very strong hunch that memory B cells, memory T cells are also having a very important role in osteoporosis. So if we can prove those memory component, and you know that the memory B cells can reside there for lifelong. So if you once we can so there should be an antigen also. So there was one report T cell memory is involved in osteoporosis. There is one report in the last 2 to 3 years that T cell memory is involved in osteoporosis. That clearly gives proves this point that there should be an antigen also. Yeah. So if that antigen has been discovered, we can call osteoporosis also as an autoimmune disease. Exactly. That itself. Right. Let's see that.
Yeah. Yeah. So there is, I can see one raised hand. Adrita, please go ahead.
Hello, Rupesh. Am I audible?
Yeah, you're audible to me. Kindly go ahead.
Yes. I saw in your slides that you have counted the number of osteoclasts with 3 nuclei, as well as the trap positive cells also you have counted. So in case of these results, how many nuclei have you considered?
More than 3 nuclei, because I have shown that it also, okay, more than 3 nuclei only we have counted that. Cells with more than 3 nuclei, we have been taken that trap positive. So because normally more than 3 nuclei is considered as osteoclastogenic, they have the potential of resorbing the bone. So we have taken only those cells, more than 3 nuclei.
Okay, thank you.
So any more questions? I'm audible or I'm online or?
Yeah.
Good afternoon, sir.
Yeah, please.
Good afternoon.
Yeah, sir, this is Vandana from PGI Chandigarh.
Yeah, Vandana.
Yes, sir. Sir, I just want to know how these cells play a role in memory cells. Just this is my query.
Which cells? Which cells?
Like Tregs and Breg cells in osteoclastogenesis, sir.
So there is only one report. I'm not remembering the name of the group. In the last, I think 2 years only, or maybe a 2020 paper, it was there that a low-grade inflammation in the body in the case of osteoporosis, there is constantly a low-grade inflammation is there, and that leads to certain T cell memory component also. There are markers for memory. They are effector central memory, effector memories are also there, which we normally look into where infectious diseases or other autoimmune diseases. But till now, there haven't been a single report which has basically proven this that osteoporosis is also having a central memory component and an effector memory component, because we still don't know whether this is an autoimmune disease or not. But I strongly feel that almost all of the diseases are having a strong immune component, and osteoporosis is one of them. And the day we are able to discover that antigen, I was already talking with the Dr. Nabed also, we will be having a strong impetus and a strong arsenal also. Again, if the antigen is there, we can do some in vitro experiments to create these memory T cells in the lab and memory B cells also, because we are having the antigen. Antigen is basically required because memory against what? Against a certain antigen. So if we have those antigens, clear-cut antigens, then only we can be able to assess or quantitate what is the amount of memory, what is the quality of the memory, and how those memory can persist for a lifetime or some time periods, and how we can tweak those memory cells, or maybe we can develop a vaccine also for osteoporosis. So these are just for right now, we don't know, but yes, in future, maybe it would be possible. It would be able to define a clear-cut antigen for this. But right now, I would say no, we still just have one paper only. So we still have to wait for that. Some other people are doing that, or we are doing that, somebody.
Thank you, sir.
Thank you.
Any more question? If there is no more question, then I will thank Rupesh for this wonderful talk. It was very illuminating talk. I'm sure we all have learned a lot from this talk. It is a very productive 1 hour exercise for learning, and we look forward to seeing your success in proving that it is osteoporosis is a autoimmune disease. That is a very provocative idea, and
Yes,
it is worth pursuing, and our best wishes to you.
Yeah, thank you, sir. I'm trying my best we can have something like this so that we can take care of this osteoporosis also well before time, developing a vaccine, vaccinating, so reducing the economic burden and mortality and morbidity of patients. Right. Yeah. Thank you. Thanks a lot, sir.
Sure. And so this concludes this seminar. We'll see you in 2 weeks' time. You will get the notification soon. Thank you.
Thank you. Yeah. Thank you, sir. Yeah.
Bye.
Bye.